I was so excited to see that there's a reproductive health section in this conference. You know, women's health needs a lot of research, so I'm very excited, and thank you so much for allowing me to come speak today on this very important topic. Let me make sure I can get this going. Okay, yes. So I'm going to be using the pointer on this screen, just so if you want to follow along. All right. So throughout gestation, multiple intrauterine organs develop alongside the growing fetus to allow it to support and grow and mature. But closer to term, around 40 weeks gestation, this is when fetal and maternal signals will increase the inflammatory load, lead to immune cell activation. This will weaken the fetal membranes that you see there in yellow, allow for cervical ripening and myometrial contractions, and eventually happy delivery of the baby. Now, these systems are totally normal and happen physiologically at 40 weeks, but they can be induced pathologically before 37 weeks, and this is called preterm birth. Now, preterm birth is a global crisis. 1 out of every 10 babies is born premature. And even in 2021, the rates of preterm birth in the United States are on the rise. Now, one of the most common phenotypes that induces preterm birth is called intrauterine infection. And this is when bacteria ascends from the vaginal cavity, crosses through the cervix, transports through the fetal membranes, and reaches the amniotic cavity where the baby will be sitting. And so this infection, as it propagates, also increases inflammation and reactive oxygen species that then leads to almost 40% of preterm births. But regardless of the pathology that induces preterm birth, these premature babies now have increased risk for adult onset diseases such as neuro or cardiovascular disease, early onset asthma, diabetes, and even certain types of cancers. And these topics might sound familiar to you because a lot of you are studying them on chip now, but they could be pre-programmed in these babies in utero. Now, during gestation, there are 2 organs in particular, the placenta and the fetal membranes, that play very vital roles as the fetal-maternal interface. And while the placenta is critical for fetal survival and growth, the membranes are also very important in the role that they play to induce labor signaling. So because of this, the fetal membranes are going to be the topic of my talk today. Now, the membranes are comprised of multiple cellular and collagen layers. This is not a simple tissue that just surrounds the baby in the intrauterine cavity. The tissue is comprised of an amnion epithelial layer that contains amnion epithelial cells, or AECs, and a chorion trophoblast layer that contains chorion trophoblast cells, or CTCs. And these 2 epithelial layers are separated by multiple collagen layers that contain mesenchymal cells, or AMCs. Now, these 3 fetal layers are connected to the maternal decidua that used to be the endometrium. And these maternal DEC cells connected to the fetal cells create the fetal-maternal interface. Now, during gestation, the fetal membranes grow and they undergo constant remodeling to maintain that structural integrity of the intrauterine cavity. And they play very important roles on the immune, endocrine, and mechanical functions that are required to support a pregnancy. Now, in particular, the amnion layers, these first 2 layers of the fetal membranes, play key roles in the mechanical integrity by undergoing cellular transitions, such as epithelial to mesenchymal transition, or the reverse, mesenchymal to epithelial transition. So they remodel the collagen, they strengthen it, they fill gaps, they maintain that barrier that's needed. While the chorion decidua interface, on the other hand, that lower layers, they play very important roles in immune regulation. They have high expressions of human leukocyte antigens that keep maternal immune cells at bay during gestation. But at term, what we know is that these 2 processes go awry. You have increased EMT, or epithelial to mesenchymal transition in the amnion that degrades the collagen and weakens the membrane. And you have a down regulation of HLA-Gs that now allow for the maternal immune cells to infiltrate through the tissue, increasing that inflammatory load, promoting those labor signals. But how do we study these types of processes in the lab? Now, traditionally, you might start off with just a single 2D cell culture system to test a method or an idea. If you're lucky and you're connected to a hospital, maybe you can collect some biopsies or explants and study the tissue ex vivo. But in tradition, you have to go to a small or large animal study to really see what's going on at the fetal-maternal interface. But there's a problem with this. And the problem is that the fetal-maternal interface has a very different function and anatomy in animals versus humans. So what we really need is an advanced human-based cell culture system that can fill this gap. And for us, that is organ on chips. So today, I'm going to tell you about our most advanced organ on chip that recreates this fetal-maternal interface, or the FMI-OC. So our FMI-OC device is comprised of PDMS, and it contains 4 concentric cell culture chambers that are connected by arrays of microchannels. This also contains an on-top gravity-fed media reservoir for long-term culture. Now, we have recreated every collagen and cellular layer of the fetal-maternal interface within this device, even going down to the detail of filling the microchannels between the amnion and chorion with basement membrane collagen, or allowing the microchannels to be left open, as you see here, so that the cells will form that nice, tight cell junction interface between the chorion and decidua. Now, within each of these circular chambers, we're able to grow primary cells that we've isolated from cesarean deliveries of the fetal membranes, and then we've immortalized them and characterized them so that we have set established cells for these chips. These cells are grown in a mixture of 2D and 3D cultures. We have AECs in a monolayer. We have AMCs and CTCs in a 3D growth that we culture with primary collagen and matrigel. And we have maternal DECs in the center that's also in a monolayer. Now, these human cells can maintain their morphology, their collagen production, seen here in purple, their intermediate filament expression, like vimentin and cytokeratin, and also their viability on chip. And since we've previously validated this device for its physiological model, in this talk today, I want to focus on how we're advancing this system to study pathologies. So our 2 objectives for this project was to, 1, model ascending infection. And so that is when you have a maternal treatment and we're looking for a fetal response. And the second objective is to recreate intraamniotic infection. And that's when you have a fetal infection and you look for the maternal response. So to start off our objective 1, we were able to establish culture within this device by loading our microchannels with matrigel the previous day and allowing it to cure. On day 2, we load our cell and collagen mixtures into the appropriate channels and fill the media reservoirs. Then, to model ascending infection, we use lipopolysaccharide, or LPS, to mimic infection on chip, and we treat 100 nanograms per milliliter into the center maternal decidua chamber. We then monitor LPS propagation, cellular level changes, and inflammatory profiles over a 72-hour period. Immunocytochemistry confirmed that LPS propagated from the maternal decidua chamber to the amnion mesenchymal chamber within 48 hours, as seen by these little red dots. And they also reached the fetal epithelial chamber within 72 hours, showing a propagation of infection from the maternal to the fetal side. Additionally, we looked at a prominent LPS signaler receptor, toll-like receptor 4, and saw that it too followed that same LPS propagation timeline as seen in this green fluorescent staining as it propagates from the maternal to the fetal compartments and quantified in this graph. Next, we wanted to look at cellular level changes. And so what we noted is that maternal LPS treatment did not induce cellular transitions like EMT in the amnion, as noted by the static levels of vimentin and cytokeratin ratios, but it did significantly downregulate HLA-G expression in the chorion after 72 hours. And so this shows us that maybe maternal LPS treatment could be inducing immune dysregulation at that choriodecidua interface. Next, we wanted to determine what maternal LPS would induce in the inflammatory profile across our device. And what we noted is a very cell and time-specific cytokine production that created a pro-inflammatory environment of interleukin-6 and IL-8, and GM-CSF across this device, starting in the maternal side and also all the way to the fetal side. So in summary for this objective, we were able to document that this infectious agent can propagate from the maternal to the fetal compartment and that it induces a fetal inflammatory response, such as increased TLR4 and downregulated HLA-G. We also saw that this inflammation was not associated with cellular level changes in the amnion. Next, we focused on our second model. This is a model of intra-amniotic infection. So we established culture on this device in the same manner as objective 1, but now we're treating lipopolysaccharide in the outer AEC chamber. We monitored LPS propagation and cellular level changes and inflammation over 72 hours. Immunocytochemistry confirmed that the fetal LPS traveled to the CTC chamber within 48 hours and to the maternal DEC chamber within 72 hours, showing infection propagating from the fetal to the maternal side. Additionally, TLR4 expression followed this LPS propagation in a time-dependent manner seen by this green staining here. When we focused on the cellular level changes, we noticed this time that fetal LPS treatment did significantly induce EMT in the amnion layers, as noted by the significant increase in vimentin to cytokeratin ratio and increased mesenchymal morphology in the amnion. But it did not change the levels of HLAs in the choriodecidua layers, suggesting that this fetal LPS could induce cellular transitions and weaken the membranes. Next, we wanted to see how fetal LPS would induce an inflammatory profile across this device. We noticed time and cell-specific cytokine productions, and in this case, specifically IL-8 and interleukin-6, that was present in the fetal amnion layer as well as the maternal decidua layer. And this was over a 72-hour time point, showing propagation from the fetal to the maternal chambers. So in summary for this objective, we were able to show that infectious agents can propagate from the fetal to the maternal chamber of our chip, and that this induces maternal inflammatory responses such as TLR4 and cellular transitions in the amnion layer, but it did not affect HLA-G expression in the chorion decidua. So lastly, we decided to validate this FMI-OC model by conducting in vivo mice testing to go along with our ascending infection model of our FMI-OC. So to do this, we treated CD1 mice on day 15 of pregnancy vaginally with live E. coli with 10 to the 11th power colony-forming units, and we monitored the ascension of this live E. coli through the uterine horns from the proxial to distal horn over 6 and 24 hours using IVUS imaging. Here you can see the IVUS imaging showing that after 6 hours, the DID stained E. coli was propagating from the proxial to distal horn, and by 24 hours, it had completely covered that horn. And along with this, when we looked at the fetal membranes, what's interesting is that these membranes at the proxial horn, the closer horn, were showing increased TLR4 and also phospho-NF-kappa B signaling, which led to inflammation and preterm birth in this model, deemed by delivery before day 18.5. So these pathways are the same pathways we're seeing in our ascended infection model on CHIP of TLR4 inflammation and weakening of the membrane leading to preterm birth. So in conclusion, we have now developed this FMI-OC, and we're able to recreate a healthy and diseased phenotypes of pregnancy. And we're able to do this because we have these well-characterized, genetically stable cell lines that we can culture within this chip, and they can maintain their intracellular interactions and provide distinct responses that allow us to make these different states. Additionally, we've conducted in vivo testing to validate this FMI-OC model and to try to prove its utility to model in utero events. And hopefully this will allow for a decrease in animal models and studies in the future. Lastly, since we have these two types of disease, the physiological and pathological states of this chip, we'd like to start to conduct preclinical testing of various agents in the hope that we can reduce these adverse pregnancy outcomes. Now, there are some limitations with our device. We don't have resident immune cells within the decidua of our chip, but we are actively pursuing to add immune cell components to this device. And as of now, we are using a static gravity-fed media reservoir instead of dynamic flow. So these are some things that we are pursuing and looking into advancing our model with. And so with that, I would like to thank the multiple labs that and the people that have contributed to this project. This project is a collaboration with Dr. Menon's lab at UTMB and Dr. Hahn's lab at Texas A&M. And specifically, I'd like to thank the people in red that have contributed majorly to this project. Also, NCATS for a good chunk of the funding for this is part of our UG3 section of that grant, NICHD, and other small pilot projects. And with that, I will take any questions, and thank you for listening.
Great, thanks, Lauren. Fantastic talk. Can we have some questions come up here? Here comes Dr. Griffin.
Hi, it's wonderful to see work on the fetal-maternal interface at this meeting. So congratulations on a great talk. One of the things we struggle with a lot in working with these models in vitro is how to manage the steroid hormones, particularly progesterone, which is very rapidly metabolized. So we tend to often use MPA, which I'm not super happy about. But the other thing is that in experiments where we use PDMS, such as Ellen Kahn, the student who gave a talk earlier, we have to work really hard to think about the design of the experiment because progesterone just soaks into PDMS like there's no tomorrow.
Okay.
And so I'm wondering, because these issues with preterm birth are often associated with progesterone resistant phenotype, which comes with inflammation. How are you managing to grapple with that progesterone issue in your cultures? Because progesterone is such a huge part of the pregnancy, of the decidua.
That's such a great question. And so I think that we're able to actually slightly overcome that in a couple of ways. Since our cells are primary derived, we've actually measured their production of progesterone naturally. They produce a lot of progesterone, specifically the chorion and amnion layer, continually throughout their cycle as a cell. And I, from being at this conference, we now know a lot of the limitations of PDMS, and I think that we will try to get away from that. But we are seeing that, the cells are functioning the way that we're expecting. And I do know that there's a lot of kind of controversy on, like, do you need to add progesterone to prevent preterm birth, or, can you use these treatments? We've also detected the progesterone receptors, the membrane progesterone receptors in these cell layers, and to document that they're not decreasing, and we know what they should do during oxidative stress. So with your concern, I think that we will go back and validate and make sure that those progesterone receptors are maintaining the right characteristics and expression on chip to make sure the progesterone is acting normally in this system. So I thank you for that comment.
It's a really hard problem, and we can talk about it later because it's really gnarly, and there's a lot of characterization you have to do. So we can continue discussions throughout other conferences too, because it's an ongoing challenge.
Yeah. Thank you for that. Yes.